Good morning, everyone. Today we will see C3, C4 and chem plants. What are the biochemistry and what is the difference between these, that has to understand. Okay, so this is the older one topics, but we have to see how it works in the different environments. For example, C3, C4, chem, crystallization acid metabolism are the three different processes that plant uses these to fix carbon dioxide. During this process of photosynthesis, fixing carbon is the way plants remove the carbon from the atmospheric carbon dioxide and it turns into organic molecules, that is called carbohydrates. Now I will give you a certain examples as well as the differences between C3 and C4. C3 plant always CO2 fix take place in the mesophyll cells, whereas C4 plants CO2 fix takes place mesophyll and bundle seed cell both. Another C3 plant also work as a CO2 acceptors is RuBP only, whereas in case of C4 plant, phosphoenol pyruvate in the mesophyll cell and RuBP in the bundle seed cell. Now what will be the C3 plant, the first product, and C4 plant first product? First product of C3, that is phosphoglyceric acid, and C4, that is oxaloacetate. This is the main difference between how it called as C3 and C4. The carbon unit is present in phosphoglyceric acid is 3, whereas in case of oxaloacetate, that is 4. Cranch anatomy is not present in C3 plant, whereas in case of C4 plant, it is present. What? Cranch anatomy. Now granum is present only in the mesophyll cells, whereas in C4 plant, granum present in mesophyll cells and absent in bundle seed cell. Okay. Now what will about the chloroplast? The normal chloroplasts are found in the C3 plants, whereas in case of C4 plant, you will observe dimorphic chloroplast. Now what is the temperature requirement of C3 and C4? The optimum temperature is 20 to 25 degrees centigrade for C3 plant, whereas in case of C4 plant it is much higher, that is 30 to 45 degrees centigrade. Now what will be how it fixations capacity? Fixation of CO2 at 50 ppm was observed in C3 plant, whereas in case of C4 plant, fixation of CO2 even less than this, means at 10 ppm it can also fix CO2. Okay. Now what about the photorespiration reactions in C3 and C4 plant? Less efficient due to higher photorespiration in the C3 plant, whereas in C4 plant you will find a more efficient due to less photorespiration. Now the enzyme. Which enzyme is useful and important for C3 plants? RuBP carboxylase enzyme used for fixations, whereas in case of C4 plant, PP carboxylase and RuBP carboxylase both are used. ATP Now the synthesis of ATP, you have to remember 8 ATP used to synthesize one glucose molecule in the C3 plant. Whereas in case of C4 plants, it consumes 30 ATP to produce one glucose molecules. Now, how efficient of CO2? Efficient at low CO2 in C3 plant, whereas in C4 plant, it is efficient at higher CO2. Now we will understand how this is happening. So C3 photosynthesis only use the Kelvin cycle for fixing CO2, carbon dioxide, that is catalyzed by ribulose 1,5-biphosphate carboxylase. This is the full form of Rubisco oxygenase, okay, which take place inside of chloroplast in the mesophyll cells. This is the reaction. Now EC number is very much important when you are writing a paper of Rubisco. This EC number is designated 4.1, 1.39. This catalyzes two different reactions with CO2 and O2 respectively in photosynthesis and photorespiration. Now RuBP plus CO2 plus H2O, that will gives your 2 PGA. Okay. Then RuBP plus CO2 plus H2O. As I said, same thing. But RuBP plus O2, that will gives you a PGA, and PGCA RuBP plus O2, that will give you a PGA plus PGCA, phosphoglyceric acid. There is a constant ratio between carboxylation and oxygenation under the special partial pressure of CO2 and O2 in the environment. Okay, so this is about C3. Now I will tell you what is happening and what biochemistry is behind the C4 plant. For example, maize plant. C4 plants such as maize, NADP, ME subtypes. Photosynthetic activity are partitioning between mesophyll and bundle seed that are anatomically and biochemically, that is, distinct. Initial carbon fixations is catalyzed by, as I said previously, that is PP, phosphoenol pyruvate carboxylase, PEPC, forming oxaloacetate from CO2 and PEP. Now oxaloacetate is metabolized into malate and then diffused into a bundle seed cell, where it is decarboxylated to provide increased concentration of CO2 around Rubisco. Finally, initial substrate of C4 cycle, PEP, is regenerated in mesophyll cell by PPDK. Now what mean by PPDK? Pyruvate orthophosphate dikinase. Generally in C4 plant, PP carboxylase, this is the EC number, perform the initial fixation of atmospheric CO2 in photosynthesis reaction, which catalyze the PEP, carboxylation of PEP in reaction that yield oxaloacetate and inorganic phosphate. This is the reaction you will find. Okay. This is the reaction you will find. Now C4 plant cycles, same thing as I am repeating. C4 photosynthesis cycle supercharge photosynthesis by concentrating CO2 and ribulose biphosphate carboxylase and significantly reduce the oxidation reaction. Therefore, engineering C4 features into C3 has been suggested as a feasible way to increase photosynthesis and yield of C3 plant, that is, for example, rice, wheat and potato, to identify possible transition from C3 to C4 the systematic comparison of metabolism is necessary. So I will tell you how it is formed. Okay. Now, the first reaction, photorespiration. Many people are think that is wasteful pathways or that occur when the Calvin cycle Rubisco act on oxygen rather than CO2. C3 plant, majority of the plant are C3, as I say, which have no special features to combat photorespiration. Whereas in C4 plant, minimize the photorespiration by separating initial CO2 fixation and the Calvin cycle in space, performing these steps in different cell types. Crystallization acid metabolism plants minimize photorespiration, save water by separating these steps in day and night time. Okay. Now Calvin cycle uses RuBP carboxylase to feed CO2 to RuBP 3 compound, that is 3GP. Okay, and that plant that fix carbon dioxide directly from the air are called C3 plant, and Rubisco can also use oxygen and alternative substrate to undergo the series of reaction known as photorespiration, and it creates a product that can be used to make a sugar, hence reduce the efficiency of Calvin cycle, and this photosynthesis by up to 25% in the C3 plant, reducing the energy yield. Okay, this is all about why. Because oxygen acts as a competitive inhibitors for Rubisco. Photosynthesis in C3 is reduced in the presence of oxygen. Now C3 plant are less efficient in hot and dry regions, as stomata must remain closed to prevent excessive water loss. When stomata are closed, O2 cannot diffuse out of the leaf, increasing O2 concentration relatively to CO2. So, in this hot and arid condition, other types of plant have evolved to limit the exposure of this Rubisco to oxygen. C4 and Chem plant uses the enzyme PEP carboxylate to combat CO2 to 3 carbon compound PEP and make 4 carbon compound. Fourth one is PEP carboxylase has a higher affinity for CO2 than the Rubisco and doesn't bind to oxygen at all. This plant can then transfer to CO2 to region with low oxygen concentrations. C4 plant, C4 pathways. Carbon dioxide is physically separated from the oxygen in order to improve CO2 binding to Rubisco. CO2 is converted to 4 carbon compound and then sequestrated to deeper tissue layer when less oxygen is present. In this deeper tissues layer, the CO2 is released, can enter the Calvin cycle without competition from oxygen. And in Chem pathway, carbon dioxide reserve, in order to improve CO2 binding to Rubisco, Chem plant adapt to arid environments where water loss is high. Stomata must remain closed during the day. The CO2 is converted into a 4 carbon compound during the night when stomata are open and CO2 is able to diffuse into leaf. This allows reserves of CO2 to be created for using during the day when stomata are closed and O2 cannot be released. Now, I will give you later on pathways of these three. and its differences.